curriculum vitae of Mr. Tommy. Mr. Tonny Cortis Maigoda is one of lecturer at Bengkulu Politeknik Kesehatan Kementerian Kesehatan Bengkulu. He has received his bachelor degree in community nutrition Universitas Indonesia and in 1996, quite long time ago. Then he pursued his master degree in interprofessional health and community care at Bournemouth University in UK and in 2016 he graduated from doctoral program in human nutrition at Human Ecology Faculty Institute Pertanian Bogor. Mr. Tonny Cortis Maigoda has pursued some training in scientific meetings and conference. He also did some scientific publication, mainly about the use of medicinal plants. He got several managerial experience at Nutrition Department and Nutritionist Association, and formerly Mr. Tonny was head of Nutrition Department at Poltekkes Kemenkes Bengkulu. Now allow me to welcome Mr. Tonny to deliver his presentation. Mr. Tonny, time is yours.
Thank you very much. Do you can hear my sounds?
Yes, we can hear your voice.
Okay, thank you. Assalamualaikum warahmatullahi wabarakatuh. Good morning. Thanks for giving me an opportunity to deliver my presentation entitled The Roles of Probiotic Supplementation Improving Gut Microbiota in Stunting Children. Okay. I would like to convey a brief outline of my presentation. This one. Okay. This is my brief curriculum vitae has been done to convey by our moderator. Okay, let's start from the global situation of stunting. Yeah, UNICEF, WHO, World Bank Group Joint Malnutrition reported that was about 149 million children stunted globally in 2020. Now, how about in Asia? According to Fanta, third project mentioned that it's about 5 to 10 million stunted children in 11 countries in Asia, including Indonesia. So according to the prevalence of stunted children in Indonesia in 2021, the highest percentage of stunted children spread out in Aceh, West Nusa Tenggara, East Nusa Tenggara, South Kalimantan, West Sulawesi, Southeast Sulawesi, West Nusa Tenggara, and East Nusa Tenggara, which were 30 to 39%. Now, look at this picture. It is very interesting picture. There are two children on the same age, but with different height. A child on the left is using school uniform. You can see that. But another one is none. Therefore, stunting is not merely about short stature. It's not merely about short stature performance, but is also regarding cognitive capacity impairment as well. This is very important. So, we talk about the consequences. The consequences of malnutrition, especially stunted, due to short-term impact and long-term impact. So, therefore, early diagnosis and prompt treatment of stunting children can avoid from these consequences. It is very very important hard effort we will do in our nations. Okay, first of all, I would like to focus on gut microbiota. What is microbiota? Scientists define microbiota as the assemblage of microorganisms or bacteria, archaea, eukaryote, and viruses present in a defined environment. So the term of microbiota is thus preceded by the name of the environment in which it is located. So, for example, gut microbiota refers to the microbiota in the intestinal tract. So, the largest microbiota of the human gut is found in the gastrointestinal tract, with about from 10 to the power of 13 to 10 to the power of 14 microorganisms. There are at least 4 functions of the gut microbiota in the body, such as harvesting energy from digestive food, secondly, protecting against pathogen, thirdly, regulating immune function, and lastly, strengthening biochemical barriers of the gut and intestine. Okay. The gut of microbiota is a complex ecological community that influences host physiology and susceptibility to disease through metabolic activity and interaction with many systems and functions of the host. So, if alteration of the GM caused either by environmental and host-related factor can impair its ability to maintain good health and lead to dysbiosis. So, dysbiosis is defined as imbalanced situation in the gut, between gut microbiota and the host is characterized by a reduction in microbial diversity and or increase for pro-inflammatory species, and has been associated with a plethora gastrointestinal extra-intestinal disorders, such as inflammatory bowel disease is very well known, IBD, obesity, cancer, and malnutrition, as well neurological disorder. So I can conclude that dysbiosis reduced diversity in the gut microbial community, or microbial immaturity are involved in several intestinal and metabolic diseases that affect growth. It is essential that an intense increase in the body size, especially in infancy, occur in parallel with the dynamic succession of gut microbiota. So look at this, is a very current article from PLOS ONE. The author concluded that the gut microbiota has been shown to play a role in energy metabolism of the host. And dysbiosis of the gut microbiota may predispose to obesity on the one hand, but in the other hand is stunting. The gut microbiota has close links to the food digestion, absorption, and intestinal function. So, persistent undernutrition in childhood will alter the normal or healthy composition of the intestinal of the gut microbiota and leading to dysbiosis. Vice versa, gut microbiota dysbiosis is associated with malnutrition and reduced plasma essential amino acid levels So metabolite produced by the gut microbiota had been shown to play a role in weight regulation. Particularly, the short chain fatty acids. Also, in stunting, an altered gut microbiota is linked to pathophysiology of stunting. This alteration may even be detected prior to actually observing stunting growth between 6 and 23 months of life. This is an existing condition, the gut microbiota in malnutrition children. I want to get a summary for these charts. Changes in the gut microbiota associated with the childhood malnutrition and key factor linked with such changes. You can see the red arrow. Yeah. Red arrow rows indicate factor that increase or decrease during childhood malnutrition. Okay, the shape of gut microbiota in malnourished children is unstable due to dysbiosis because the number of commensal is a good microbiota, gut microbiota is falling down. And the pathogen microbiota is going up. This is unbalanced situation. Make it, the digest cannot work properly. So how the children can get essential nutrition in order to grow and to develop? This is the problem. So what factor influence the gut microbiota in early life? Gut microbiota vary by term of births, mode of birth, using oral antibiotics, mode of feeding, maternal diets, presence of fewer fats, host genetics, presence of sibling, and family members, complementary diet, and local environment, such as urban versus rural and geographical location population. Mode of milk feeding and complementary diet has been defined as the strongest determinants of infant gut microbiota composition as indicated by the arrow size. You can see that. Impaired development of the gut microbiota causes stunted children to have an immature gut microbiota as compared with children on the same age. So we talk about the maturation of the gut in microbiota. Maturation of the gut microbiota, the composition of the gut microbiota in children varies greatly during the first years of life before reaching a more stable and mature composition. So maturation in adult state is due to several factors such as genetics, environment, gut physiology, and diet. So, in addition, mode of feeding influences early infancy microbiota and saccharolytic versus proteolytic fermentation in the gut with potential mechanisms, potassium mechanism in the gut. A breast milk contains human milk oligosaccharides, which human ingestion by the infant pass undigested through the upper gastrointestinal tract until they reach the colon. Here, they are digested by specific HMO, degrading bifidobacterium species into various metabolites with potential metabolic, with potential beneficial health effect. This is A, this is B. And B, if the baby using formulated milk, yeah, this is condition. Formula milk contain excess protein, some of which human injection is incompletely digested and absorbed in the upper GI tract and reaches the colon. Partly digested protein, peptides, and individual amino acids are metabolic by gut microbiome, including opportunistic pathogenetic bacteria, such as Clostridium and Enterobacterias species, and yielding various amino acid catabolites, some of which have potential detrimental health effects. It is a serious problem as well. Okay, selected mechanism through which gut microbes affect the process of growth in children. The mediating role of the intestinal microbiota in childhood consists of participation in metabolic process through the production of biologically active metabolites, mainly short-chain fatty acid. In the metabolism of human milk and bile acids, participation in the hormonal activity of the host through indirect impact of the hormone levels, including insulin-like growth factor, peptide, glucagon-like peptide, and serotonin, and stimulation of the innate immune system, as well as initiation of the inflammatory process. So this is early colonization of the gastrointestinal tract in newborn. Yeah. Early colonizer of the gastrointestinal tract are specific to the type of delivery. In the case of vaginal delivery, early colonizer originate from the mother's vaginal and fecal microbiota, whereas for caesar section, yeah, the mother should take an operation. Early colonizer belong to the environment of birth and mother skin microbiota. Breast milk microbiota also colonizer the gastrointestinal of newborn, according to Jenny Allo 2016. So there is an interesting review article written by Ruari in 2019, mentioned that window of opportunity to enhance growth may also exist beyond the first 1,000 days. The assembly and maturation of the gut microbiota has largely occurred by the age of 2 or 3 years. Hence, strategy to target the gut microbiota after this period may have less impact. Yes. However, the periconceptional period in adults and female beginning 14 weeks prior to conception may represent an additional window of opportunity in which to optimize nutrition and the gut microbiota, which may have significant benefits for health and nutrition behaviors, suggesting potential for microbiota targeted therapy as well. You can see these graphs, yeah. This is the different pathway, yeah, of healthy and undernourished growth. Yeah. Current evidence suggests that a number of bacterial signatures are associated with either undernutrition or healthy growth during the first 1,000 days. During pregnancy, you can see here, yeah, a vaginal microbiota with long diversity, and rich in lactobacillus is associated with term birth and normal birth weight in high-income setting countries. However, healthy growth is associated with greater bifidobacterium lojum and Streptococcus thermophilus in the first 6 months of life, which are less prevalent in early life undernutrition. All about breastfeeding. Breastfeeding during this period is associated with a greater bacteroid and bifidobacterium with healthy growth, while Escherichia coli, Staphylococcus aureus, and other species are associated with severe acute malnutrition. We have to be careful with this. This is mechanism of microbes in the intestine lumen interact with growth. The commensal gut, it is a good microbiota, regulate a number of processes that affect child growth in the first 1,000 days. The structural and the functional integrity of the intestinal barriers, mucus layer, antimicrobial peptide, and epithelium and tight junctions is tightly regulated by gut microbial composition in healthy infant, but become perturbed in undernutrition. A healthy microbiome also provides colonization resistance against invading pathogen. The interaction between the commensal microbiome and the innate immune system maintain immune homeostasis and recognition of antigens. So the microbiome plays a critical role in nutrient and host metabolism, thereby affecting digestion, absorption, and energy storage. A dysbiosis, on the other hand, microbiome in early life may impair each of the pathways related to growth, whereby an immature microbiota fail to protect the intestinal barrier, leading to various blunting, mucus degradation, intestinal permeability, impair immune response. Okay. This is a complementary feeding increase gut microbial diversity and production branched chain fatty acid and with potential implication for growth, neurobone, and immune development. So complementary feeding increase gut microbial diversity and production of branched short chain fatty acid and short chain fatty acid with potential implication for growth, neuro, bone, and immune development. This is the whole integrated system. As complementary feeding progress, the milk-based component is gradually replaced by other foods such as meats and dairy, fruits and vegetables, and bread and cereal, which are directly, possibly indirectly, impacting growth and development. These foods contain protein and dietary fiber that modulate the infant gut microbiota, increasing alpha diversity and the abundance of key bacterial families that produce short chain fatty acid, not that excited is also produced in high amount during early infancy. And brain chain fatty acid during complementary feeding is also important. So this is a conceptual model describing how the maternal gut microbiome may mediate effect of malnutrition on early neurodevelopment. Yeah. Panel one, this is panel one. Yeah. Both over and undernutrition and alteration in ratios of macro or micronutrient lead to perturbation in the maternal microbiome during gestation. This perturbation include alteration in diversity and relative abundance of specific taxa. Panel 2. Changes in maternal nutrition affect circulating biomolecules, immune homeostasis, and endocrine function. So, these changes may be mediated by observed shift in the maternal microbiome. Lastly, in panel 3, these changes in maternal biology can in turn directly or indirectly impact the fetus, including developing fetal brain. So, impair development of the gut microbiota causes stunted children to have an immature gut microbiota as compared with non-stunted children on the same age. The evidence of causal role of the gut microbiota in delayed growth is increasing. However, an immature and less diverse microbiota, which is often present in children with delayed growth, may reduce or prevent the success of nutritional intervention aiming to promote growth. Therefore, in recent decades, the scientific focus has shifted to intervention targeting the gut microbiota such as probiotics, prebiotics, and symbiotics because they may have the potential to address stunting effectively. So I will try to explain one by one the role of three functional of first one I call the probiotics. Probiotics live microorganisms which, when administered an adequate amount, confer a health benefit on the host. Probiotics are strain-specific can result different health benefits. They may improve child growth through modulation of the gut microbiota and immune system, inhibition of pathogen growth, prevention of infections, lowering diarrhea incidence, improving of the absorption of energy, and improvement the absorption of supply micronutrient. That's why the useful of nutrient can be absorbed perfectly. And the role of prebiotic is substrate that is selectively utilized by host microorganism, conferring a health benefit. Prebiotic promote to the growth of more or same one specific bacteria, mainly bifidobacteria and lactobacilli, leading to, among other things, the production of short-chain fatty acid, which inhibits pathogen growth. In addition, probiotic enhance the uptake of micronutrient, delay gastric emptying, improve gut barrier function, and modulate the immune system. And lastly, what is the role of symbiotic? Symbiotic is a mixture, mixture between probiotic and probiotic. Live microorganisms and substrate selectively utilized by host microorganism that confers a health benefit on the host. The beneficial effect of probiotic and probiotic taken separately may be enhanced further if combined. I would like to talk of scientific finding. This review article, it is very, very good article from ASN, Current Development in Nutrition. The objective of systematic literature was to evaluate the efficacy of probiotic, prebiotic, and symbiotic intervention compared with control on improving growth outcome in children living in low and middle income countries. We can see the result. This is the age, this is the place, and how long should be length of intervention. And this is a probiotic intervention. This is by using a vehicle with curd. There are three selective evaluation of this result. It's primary, secondary, or secondary. There are many studies have been done in order to test the efficacy of prebiotic, probiotic, and symbiotic. We can see that there are so many authors have been doing a lot of research regarding to the prebiotic and symbiotic treatment. Okay, you can see that here. I can read the one by one. It is many of authors have been referred. So I want to change to the main finding. After reading the whole of review article, I would like to conclude that the role of probiotics, the 11 studies gave little evidence that probiotic affect the growth of children when compared with control. But however, a higher percentage of studies, including undernourished children, showed an effect than of studies including healthy children. Probiotic may be more effective in undernourished children than in healthy children because they may restore the dysbiotic microbiota affecting growth. A limitation of the probiotic analysis was that antibiotic usage was not taken into account. This is forgettable, owing the small number of included studies. Yet, this could affect the outcomes.